Okay, hi, my name is Natasha Auer, and this is,
Rocio Vazquez-Beltran.
and we worked in the lab of Dr. Vinicio de Jesus Perez on the role of exosomal long non-coding RNAs in pulmonary arterial hypertension, or PAH.
So to give you guys a little background of what pulmonary arterial hypertension is, we know it's a disease of the pulmonary vessels. So in the larger arteries, as seen in the picture on the left, it causes some vascular remodeling and arterial wall thickening, which eventually results in right heart failure. As we can see in the picture in the right, blood vessels in our lungs become narrowed and it obstructs the blood flow. As a result, this blood flow through the pulmonary vessels decrease and the heart needs to work harder to pump this blood through, causing the right ventricle to thicken and eventually give up. In the microvasculature, like arterioles and capillaries that we see on the left, PAH causes small vessel loss.
So this small vessel loss is a result of disrupted endothelial cell and parasite cell interactions. On the left here, we see normal endothelial cell-parasite cell interactions. Endothelial cells line the lumen of the blood vessels, and in microvessels, like capillaries, parasites interact with endothelial cells to keep the vessel strong. On the right over here, you can see what can happen when these interactions aren't strong. So here you see that in a healthy donor patient, they're healthy endothelial cells and parasites. The parasites and the endothelial cells interact really well. On the very, very right here, you can see in a PAH patient that the endothelial cells in green and the parasites in red don't interact very well at all.
So we have talked to you about PAH can lead to the disruption of these endothelial parasite interactions, but we want to know more. We want to know how this actually occurs. So one possible mechanism is through exosomal LNC RNAs. So as you can see in this picture, a cell is secreting exosomes, and inside we can find this LNC RNA, which will then target another cell. Now, exosomes are extracellular vesicles that have been recently shown to contain proteins, RNA, DNA. Of interest to us are specifically the long non-coding RNAs that they actually carry. So these long non-coding RNAs are also called LNC RNAs, are a category of RNAs that are longer than 200 nucleotides in length and do not have a coding potential, but they can surely help in gene regulation by mediating gene expression. So in normal situations, these LNCs RNA are degraded fairly quick, but in abnormal situations, as in the case of PAH, this LNC RNA expression is stabilized, having them to cause a modulary role within the cell. So with this in mind, we wanted to determine if these exosomal LNC RNA cargoes from endothelial cells and from parasite cells of the patients that had PAH play a role in this endothelial parasite interaction and this long vascular function. So this is a more in-depth explanation to really drive home what LNC RNAs are and why they could be important in PAH. Here we see all the possible mechanisms of action to modulate this gene expression. So some examples are sponging, chromatin looping, and acting as a chromatin modifying enzyme. As of now, we do not know which of these mechanisms possibly contributes to the disruption of this interaction between endothelial cells and parasites, if at all. So our hypothesis remains very simple. So it is identified LNC RNAs may contribute in preventing establishment of these endothelial and parasite interactions, which then results in vessel instability as seen in PAH. So we have gotten this far with theory, and now we actually wanted to start investigating a little more.
So our first point of investigation began with an RNA-seq analysis of lincRNAs in endothelial cells and parasite exosomes. The top 5 lincRNA candidates from each of the 2 cell types were determined based on highest fold change and lowest p-value. Rocio and I's job through this 10-week program was to characterize these 10 lincRNAs as best as we could and determine our top 2 candidates, one from each cell type, to share with you today. So To do this in silico analysis of the candidate RNAs, we went through about 7 steps of many different bioinformatic websites. So we started again with the RNA-seq. From there, we then used Linkopedia, LinkRRI search, and gene cards to do our initial analysis. And once we had decided which lincRNA candidates we wanted, which top 2, we used UCSC's genome browser, MIRDB, and David to do a little bit of further analysis to really set in stone these 2 lincRNA candidates and to prepare for future in vitro studies that our lab will be doing. So our first candidate was found in the exosomes of endothelial cells and is titled LinkMax2 :1. So this lincRNA was found to be significantly upregulated in the exosomes of endothelial cells of PAH patients. When we ran our lincRRI search, we noticed, this is our lincRRI search, that it was predicted to be associated with microRNA MIR6820. Now, this was interesting to us because we found a paper that found that this microRNA was significantly upregulated in a paper that looked at hypoxic conditions. Now, this is of interest to us considering that hypoxic conditions can induce this endothelial cell parasite disruption that we were looking to better characterize, or what our whole project is really about. So we did a little bit of further analysis through MIRDB. Before we move on to that, though, I just wanted to quickly clarify that microRNA and lincRNAs are fairly similar, but microRNAs are a lot smaller than lincRNAs are, but both of them still don't have coding potential. That is what mainly makes them similar. The other thing I wanted to point out, because you may ask, why didn't we choose to characterize these 3 other genes up here? The reason is that they're found to be pretty universally associated with all the lincRNAs that we looked at, which makes us think that they're not very specific to the disease and maybe has something more to do with the role of lincRNAs in general. So now moving on to microRNA 6820. So we used MIRDB to do a little bit of a further analysis and found that MIR 6820 associates with genes involved in the regulation of endothelial parasite interactions. So of interest to us, it was the association of this microRNA with angiopoietin-like 3 and MAP4K3. The reasons these are interesting is that angiopoietin-like 3 associates with another protein called Ty2, or interacts with Ty2, a tyrosine kinase that is important in ECPC, or endothelial cell parasite interactions and vessel stabilization. It was also found to be associated with MAP4K3, which is part of the MAPK-ERK pathway, which is another pathway involved in proliferation, migration, and differentiation characteristics of PAH. So overall, we found the fact that this lincRNA was related to this microRNA pretty well, which was then linked to other genes which were important, or genes and proteins important in endothelial parasite interactions. We thought that that was a pretty good lincRNA to look at.
So the other lincRNA we looked at was link API 56, which was chosen because it was the only LNC-RNA of the top 5 in the parasite exosomes that could be well characterized enough. A limitation that we'll talk a little about in the next slide. It does also, however, have some importance beside its fold change, and is predicted to associate with ABI2, which is a gene that may act up in the regulation of cell growth by interacting with tyrosine kinase receptors, as mentioned before, which is involved with tyrosine kinases. So FAM98B was also of interest, as it seemed to be connected to MAPK pathways, which have been shown to be involved in PAH pathogenesis pathway, thus connecting to this disease. However, upon further, it seems that this might not be true. So now we have looked at our 2 LNC-RNAs of interest, and remember that our original thought was that these LNC-RNAs somehow may contribute to preventing establishment of these endothelial and parasite cells interactions, which results in the vessel instability that we see in PAH. So at this point, we have done some bioinformatics data collection, so our next steps are validation and expression studies. This picture here highlights the locus of conservation, which is a step needed to inform us about whether or not we can do studies in animals. So this is very important. Other future studies involve validation of presence or absence of LNCs, RNAs in exosomes, and we will create primers through a bioinformatic website called ensemble.org. Once this is done, the lab will perform knockout and overexpression studies in human parasites and microvascular endothelial cells in vitro, thus to assess the contribution in maintaining this endothelial and parasite behavior and interactions.
So we know we went a little bit over time, but we thank you for watching our presentation, and we wanted to give a big thank you to the Stanford CBI program, Dr. Vinicio de Jesus Perez, Dr. Stuti Agrawal, and Dr. Ananya Chakraborty, who all helped us during this project enormously. So thank you